Well, hello everybody, and welcome to Week 5, the Week 5 review video. You may hear a blowing sound in the background. My apologies, it's getting pretty cold in the basement here in the Manila household, so I have a heater going. And apparently along with the heater comes the cat, who really likes sitting in front of it, so if you happen to hear him or he walks across the board, my apologies in advance. For those of you that had me in genetics, it is Jet, our old friend with his incomplete penetrant, temperature-sensitive fur color. You might remember he was the focus of a challenge question in genetics, but yeah, he's with us here, in spirit and in person. So these are the topics that we're going to be talking about in today's review video. We've got the trip repressor. We'll use that as a small story, an example for a lot of DNA binding stories. We'll talk about DNA binding motifs quickly, and then how DNA binding proteins such as the trip repressor bind to DNA through the major groove, and that'll allow us to talk a little bit about specificity as well. We'll move on to just very quickly talk about dimers, homo and heterodimers, and how that relates to DNA binding. And we'll talk about regulation at a distance and this whole idea of looping out. A lot of you said you weren't quite sure what was meant by that looping out. Then we'll move on to coordinated regulation and combinatorial control. We will use that ROX1 story as our coordinated regulation example. We'll use mediator as our example of combinatorial control. And we'll end with a recap of RNAi, or RNA interference. So it looks like a lot, but single stories cover a lot of these topics, as we're about to see. So it's not quite as much as we think that it is. So in E. coli, or in prokaryotes in general, there are what we call operons. Operons are sets of genes under the control of a single promoter. So if this single promoter is on, a single messenger RNA is made for this entire operon. That single messenger RNA contains multiple coding regions. Ribosomes, through their Shine-Delgarno sequences, can bind upstream of those coding regions, and multiple proteins are made. And this actually allows coordinated regulation to occur in prokaryotes, because if each one of these genes is responsible for tryptophan biosynthesis, the biosynthesis of the tryptophan amino acid, each one of these genes encodes each one of these proteins that's required to make tryp, then it makes sense that there's never going to be a case where the cell only needs this protein. It's kind of like a package deal. If tryp levels are low and the cell needs to make tryp, then the cell needs all three of these proteins to make new tryptophan. If tryp levels are high, then the cell doesn't need any of them. So since the cell is always going to be, and Jet's leaving by the way, so we should be fine. So since the cell is always going to be in a state where it either needs all of these proteins or doesn't need any of them, it makes a lot of sense to have them under the control of a single promoter. So if we fire this promoter, we make this polycistronic RNA, we make all of these proteins, and we have the outcome that we want. So because of that, prokaryotic cells tend to regulate these operons by using a single regulatory factor. And in this case, that's going to be the trip repressor. From its name, we know that by a repressor, that means it's going to turn this operon off and keep these proteins from being made. In fact, it's going to keep this messenger RNA from being made. So we have to ask ourselves, when would the cell want this operon off? Remember, this operon is used to make tryptophan. So the cell needs these proteins to make tryptophan. In other words, the cell needs these proteins when tryp levels are low. When the amino acid tryptophan is in short supply, the cell needs to make more. It turns this operon on, and more tryp is made. So tryp is going to be off, or the tryp operon is going to be off, when tryp levels are high. If tryp levels are high in the cell, we don't need to make more. We already have it on hand. We can have this operon off and not make these proteins. We would repress the trip operon. So we want the trip operon repressed when trip levels are high. To say that a little bit more specifically, we want the trip repressor binding to DNA when trip levels are high. Trip levels are high, we don't want the operon on, we don't need to make more, we want repression. That means we want the repressor to bind here. So when trip levels are high, the operon is off. When trip levels are high, the repressor binds to the operon. So that's the conceptual understanding. Let's now talk a little bit more about the molecular mechanism. So what I'm doing here is representing DNA a little bit more specifically, and we have a major groove on the DNA molecule, and we have across from it the minor groove, and these flip-flop. So that means this would be the major groove of the next twist, and this would be the minor. And we're talking about the trip operon, and what I've drawn here is that upstream region I had here just a moment ago, where the trip repressor is binding. So now I'm going to put that trip repressor here in its binding configuration. So here is the trip repressor. We've just blown up that region that I had drawn here on the left just a minute ago. So the trip repressor is binding. Now, if it's binding, that means trip levels are high, and the way that the cell knows that trip levels are high is because tryptophan itself, the amino acid itself, binds to the repressor. And when tryptophan is high, when tryptophan levels are available to the cell, in other words, when the cell doesn't need the trip operon to be expressed, when trip levels are high, some of that tryptophan binds directly to this trip repressor protein, and now this protein binds to the DNA. And as I've drawn it, it binds to the DNA in the major groove. So almost all DNA binding proteins bind to the major groove of DNA, where the DNA is more open and more accessible. Or I should say the bases of the DNA are more accessible. In fact, one of the only common proteins that binds to the minor groove of DNA is TBP, Tata binding protein. And it does that so it can twist or kink the DNA. But anyway, we get the binding here. So like many DNA binding proteins, the trip repressor binds to a specific sequence of DNA. So there might be an adenine right here, a guanine here, a cytosine here, an adenine here, and a thymine here. That would make the binding site or the recognition sequence of the trip repressor AGCAT. Now I'm just making that up as a hypothetical. The trip repressor binding site is known. I'm not going to use it here just because I don't want to take the trouble to look it up. Each individual base pair has its own non-covalent bonding potentials. What do we mean by that? What we mean is that, again, for example, adenine might give off a hydrogen bond acceptor, right? So this might be a partially negative group on adenine that can accept a hydrogen bond, the way that we've described it before. For guanine, let's say for argument's sake, guanine offers a hydrophobic region of the base that's accessible in the major groove. Cytosine might be hydrogen bond donor. Now this is another adenine, so we already know that adenine is a hydrogen bond acceptor. And then we have thymine. Well, let's say that thymine has a van der Waals potential. Non-covalent force potentials, right? None of these bonds are made. These are the potential for bonds. If there's a hydrogen bond donor across from this acceptor, that can be an H bond. If there's a hydrophobic group across from this guanine, that can be a hydrophobic interaction. If there's a hydrogen bond acceptor across from this donor, that can be another. So these are all kind of like half of a puzzle piece. These are all just the potentials. Now, let's imagine that the trip repressor protein has its own non-covalent bonding potentials. And they're arranged in three-dimensional shape because for protein, structure equals function. So the three-dimensional shape of this trip repressor governs what it can do. And let's say that right here on the trip repressor, it has a hydrogen bond donator group. And right here on the trip repressor is a hydrophobic region. And right here on the trip repressor is an H bond acceptor. And right here is an H bond donor. And finally here, we have van der Waals potential. Now if you look at what I've done here, we have an H bond donor right here across from the H bond acceptor of the adenine. There's an H bond. Well, I said that there was a hydrophobic pocket on a guanine, and here's a corresponding hydrophobic region of the trip repressor. So we're going to get a nice hydrophobic interaction here. The trip repressor is accepting an H bond in this region. That lines up perfectly with our cytosine H bond donor, another hydrogen bond. another hydrogen bond here, and finally van der Waals potential on both sides of this gap. That's a van der Waals interaction. So this trip repressor mirrors or complements the non-covalent bonding potentials of the sequence it recognizes. We used the analogy of Velcro earlier in the course to talk about why one strand of DNA sticks to another, but the same logic applies here. Each one of these individual non-covalent forces is not enough to keep this trip repressor anchored to this sequence, but combined, when you get all of them across the surface, it is enough Velcro to keep the trip repressor bound. This is specificity in binding. This is how proteins bind to DNA with specificity. And we can play with this a little bit. So let's imagine that the trip repressor is trying to recognize some other sequence, maybe AGAAT. And if we look, A is an H-bond acceptor, in other words, a partially negative region. And right here, on this region of the trip repressor, we also had an H-bond acceptor. Well, if we have a partially negative region here on the trip repressor and an equally partially negative group here on this adenine, those would repel. So if we had an adenine in this one position, not only would we lose Velcro, we would actually have repulsive forces pushing the trip repressor away. We've lost our specificity. We will not have binding. So we only get the trip repressor binding to its sequence specifically when the non-covalent bonding potential of the sequence perfectly matches the non-covalent bonding potential of the DNA binding protein. And then we get that binding. Now, let's go back to the trip repressor story specifically. This is all happening when trip levels are high. So what happens when trip levels are low? Well, when trip levels are low, the trip repressor undergoes a conformational change. In other words, a shape change. And that shape change alters the DNA binding surface so that now the hydrophobic pocket is here, the hydrogen bond acceptor is here, and the hydrogen bond donor is here. We've basically pulled those non-covalent bonding potentials away from the binding surface. That means we have Velcro here, and that's it. Well, that's not enough. That's not enough non-covalent force to keep the trip repressor bound to this DNA, and so the trip repressor lets go of the trip operon. But that's wonderful, right? Because what's making the trip repressor have this configuration is that the trip amino acid is not bound to it. Well, the trip amino acid is not bound to this repressor because trip levels are low in the cell. And if trip levels are low in the cell, we don't want repression. We want the trip operon on so that the cell can make more tryptophan. All of that happens. The cell starts cranking out tryptophan. It's making tryptophan, synthesizing this amino acid. Trip levels climb. Trip levels climb so much that some of that tryptophan binds to the trip repressor and induces this conformational change. With tryptophan bound, the trip repressor goes back to this extended binding surface. Now, this structurally mirrors the bonding potential of the recognition site perfectly. All of these non-covalent bonds can be made. The Velcro is in place. The trip repressor binds to the operon and represses transcription. The cell stops making the enzymes to build tryptophan, which is great because tryptophan levels are low. It's time to stop making more, is high, and it's time to stop making more. So we're covering a lot there in this example, but in a wonderful way, right? We're talking about binding to the major groove. We're talking about binding with specificity, but we're doing it through the example of the trip repressor. So what is a DNA binding motif? A DNA binding motif is any inseparable portion of a protein that has that function, in this case DNA binding. So this, in green, is the DNA binding motif of the trip repressor. It binds to DNA, that's its function, that's its structure in the trip repressor. That makes it the motif, or the portion of the protein that does that job. So good stuff there, lots of good overall themes. Everything I said about binding with specificity doesn't just apply to the trip repressor. Again, we're using this as an example. Every single protein that binds to DNA through this kind of mechanism, this mirroring or complementing of non-covalent forces. That's the crux of all DNA binding with specificity. And before we move away from this, I just want to do a better job of showing the major and minor groove. This image does it a whole lot better, obviously, than I could ever draw. And you see here that in the major groove, the major grooves are shown in red. In the major groove of DNA, all that kind of gold or orange space-filling diagram that you see, that's all the bases. Look how much access there is to the bases there. I can get in here and touch these bases for hydrogen bond donor and acceptor groups, for hydrophobic pockets, for ionic interactions, for van der Waals interactions. Tons of room to manipulate and feel these bases and get that specificity. Compare that to the minor groove, where there's a lot less room between the backbone. The backbone of DNA is in blue here. A lot less space and room to access these bases. That's why most of these DNA binding proteins bind through the major groove. All right. Now, dimers. What is a dimer? Well, to say it most simply, a dimer is a pair of proteins or polypeptides that come together to form a larger complex. So this is a dimer. This is one polypeptide or a subunit. This is the second polypeptide or a subunit. And these two polypeptides are interacting non-covalently to make a single complex. And more specifically in this case, a single dimer, a complex made up of two individual subunits. More specifically, this would be a homodimer because I've drawn the two subunits as being identical. We can also have heterodimers. Heterodimers fit the bill of a dimer, two individual subunits, two polypeptides that are interacting with each other non-covalently to make a larger complex. And because it's made up of two and only two subunits, this is a dimer. But obviously, these two dimers, these two subunits of this dimer are different. So heterodimer means the subunits are different. Homodimer means the subunits are the same. Now, when it comes to DNA binding dimers, each subunit has its own ability to recognize a sequence of DNA, just like we talked about a moment ago for the trip repressor. These things can reach into the major groove, they can manipulate and touch the bases, they can mirror and complement those non-covalent bonding potentials, and they can bind as long as those bonding potentials complement each other properly and meet up. Let's say, for argument's sake, that the sequence that this subunit can bind to is TAGC with specificity, the way that we've said before. And this sequence represents GCAA, this subunit. What I was trying to say in the lecture, the point I was trying to get across is once you know this kind of information, you can predict the recognition sequences of these different dimers. In other words, this homodimer is going to bind to the DNA recognition sequence TAGC, TAGC. How do I know that? Because each one of these blue subunits recognizes TAGC, and since this is a homodimer, it's going to recognize TAGC twice in a row, adjacent to itself. That's the recognition site. Conversely, I know that this heterodimer is going to recognize the recognition site TAGC, GCAA. Because each of these individual subunits recognizes each of those individual sequences, but once you make a heterodimer, this heterodimer, can only bind to this specific 8 base pair sequence, TAGC, GCAA. So, by mixing and matching subunits into homo and heterodimers, the cell can get the best binding potential, the best specificity that it needs for any given function. So that's really what we were trying to talk about there in the context of dimers and how DNA binding and specificity plays into dimers. Last point on all of this. Now, in most simple prokaryotic systems, if you're going to get that kind of binding, that kind of heterodimer binding, homodimer binding, trip repressor binding, anything we've been talking about, most of the time you're going to get that binding occurring near a gene in prokaryotic systems. If this is a repressor, it's going to block RNA polymerase from binding and will repress the gene or the operon. If it's an activator, we're going to promote RNA polymerase binding and we're going to turn the gene on. Whatever it is that this dimer is supposed to be doing, it'll do it right next to the promoter, right next to the control region of the gene. However, in eukaryotic systems, that's not always the case. In eukaryotic systems, we often see the transcription factor, in this case the homodimer, we see it binding way, way far away from the actual gene and its promoter. So, if we're going to recruit the pre-initiation complex right here, remember this is the promoter in a eukaryotic system, so this is the TATA box. That means it's going to be bound by TATA binding protein, part of TF2D. TF2D is going to recruit A and B, and then F is going to come in with the polymerase, and then E, and then remember DABFA, the pre-initiation complex. Carboxy-terminal tail will be phosphorylated by H. That'll release. All of that needs to happen right here. So how is this transcription factor, this specific transcription factor, going to influence what happens so far away in space and in sequence from this gene? Well, we have to remember that DNA is flexible. And so that transcription factor can immediately interact with the pre-initiation complex. It can immediately, physically, interfere with, as a repressor, or promote, as an activator, the binding of RNA polymerase, without ever leaving its binding site. But what has to happen is that all of that intervening DNA that I had drawn in all this region, it has to loop out of the way. We've basically taken the dimer that was binding out here, and we've bent the DNA back on itself. We've looped the DNA out so that that could interact with the transcription factor. This might be better to do in 3 dimensions. So if this is the gene right here, and this is the promoter of that gene right here, and the transcription factor is binding here so far away, and we want to influence all of the proteins that bind at the promoter, what can we do? We can just bend that DNA around, and as soon as we do that, we have brought that transcription factor into the general vicinity of that promoter. So, and what we mean by looping out is all of this DNA that had to bend and loop to accommodate that interaction. So that's the regulation at a distance that we were referring to there. So how are we doing? I think we've done a good job talking about the TRP repressor. I hope you agree. We touched on DNA binding motifs. We definitely talked about DNA binding, the major groove in general, why we need access to the sequences, and then that story of specificity, how we interact with those sequences specifically to get enough Velcro to stick us there. We talked about dimers, heterodimers, and homodimers, and we talked about the looping out of DNA, regulation at a distance, especially in eukaryotic systems. This is most common in eukaryotic systems. Now we're on to the second lecture of the week, coordinated regulation and combinatorial control, before we end with RNAi. So, coordinated regulation. What does that mean? Coordinated regulation is the regulation of many different genes that are controlled by a single factor. Now that factor can be an environmental stimuli, or it can be a single transcription factor. What that factor is can be a number of different things, but the idea here is that many genes are regulated by one thing. What is combinatorial control? Combinatorial control couldn't be any more the opposite of coordinated regulation. The idea of combinatorial control is that many factors, and again, this can be environmental stimuli, environmental factors, transcription factors, many factors regulate one gene. So coordinated regulation, many genes are regulated by one thing. Combinatorial control, many things are regulating a single gene. Our example for coordinated regulation from the lecture was primarily the ROX1 story, and our example for combinatorial control is mediator. So now let's talk about those two examples in some more detail, but always keeping in the back of our mind the definitions of each. So, coordinated regulation. How are we going to control many, many genes through one factor or one signal? So a little bit of background here. Baker's yeast, also known as brewer's yeast, is a budding yeast. It divides asymmetrically, and as its common names suggest, it does two different things. It's used by bakers for bread, and it's used by brewers for beer and for wine. So we already implicitly know, just based on those names, that this interesting little single-celled eukaryotic microbe can do two completely different things. It can make ATP through respiration. And when I say respiration, I mean just like us. In respiration, this yeast takes oxygen in. Let me lower that just a little bit. It takes oxygen in and releases carbon dioxide, and that generates a lot of ATP for this little yeast cell. It's this carbon dioxide release that makes bread dough rise. When we put yeast in bread dough and we put sugar in that bread dough, we give it some time and some heat, the yeast is taking oxygen in and exhaling, really, on a cellular level, carbon dioxide, and it's those carbon dioxide bubbles that make the bread dough rise and make the bread soft. So that's respiration. Now, yeast can also ferment. And when yeast ferment, they make ATP by taking sugar in and releasing ethanol, or alcohol. This is the brewer's side of Saccharomyces cerevisiae. Now we are taking in sugar, we're making ATP inefficiently, but we're doing it in the absence of oxygen, and alcohol is the byproduct. So when yeast allows us to make beer and make wine alcoholic, it's because of this byproduct of yeast here. So, as I've alluded to, this is a much more efficient system. This is the better way to go. This is less efficient. But this respiration requires oxygen levels to be high, whereas this system is only really preferred by the cell when oxygen levels are low. I'm sure it wouldn't surprise you to know that there's a whole bunch of proteins that are needed to make this possible, and that means there's a whole bunch of genes that encode those proteins, and conversely there's a whole other set of proteins with their own genes that make fermentation possible. So if you were a yeast cell and oxygen levels were high, what would you rather do? Would you rather ferment or respire? Well, when oxygen levels are high, you'd rather respire. So you want to repress your fermentation genes and express your respiration genes. This is the preferred system. When oxygen levels are high, breathe. Make ATP through respiration. Oh, he's back if you can't hear him, and he's right here. When oxygen levels are... Come on, let's go. Excuse me. Sorry about that. When oxygen levels are high, the cell is going to respire, so it wants to express these genes and repress these genes. When oxygen levels are low, we want the opposite outcome. We want to express our fermentation genes and repress our respiration genes. We can't respire when oxygen levels are low. This is not an option. So although it's the preferred mechanism, it's impossible in low oxygen. So we repress these proteins. We don't make these proteins because we can't use them anyway. And we express the least preferred system because that's the only system the cell can survive on. It's the only system they can use to make ATP. So that's the conceptual understanding of all of this that we need to go into this story of coordinated regulation and understand what's happening. So we are going to focus on the fermentation genes. Remember, for the fermentation genes, we want them off when oxygen levels are high. When oxygen is high, we'd rather breathe. When oxygen is high, a yeast cell would rather respire. We only want these genes on when oxygen levels are low. So that means the cell requires a mechanism by which it can measure oxygen availability. When oxygen levels are high, the cell has the ability to make heme. When oxygen levels are low, the cell cannot make heme. Heme biosynthesis requires oxygen in 2 steps, so cells can only make heme when oxygen levels are high. When heme is made, it complexes with a protein called HAP1. In yeast, it's great because everything is named for what it does. HAP, H-A-P, stands for heme-activated protein. So what do you think heme does to HAP1 when it binds to it? It activates it. So this heme-HAP1 complex is now active, and it is an activator for a gene called ROX1. Now the cell makes the ROX1 protein. Let's take a quick break and back up. Why did we make ROX1? Well, we made ROX1 protein because the ROX1 gene is active. Why is the ROX1 gene active? Because the HAP activator is on. Why is the HAP activator on? Because heme is present. Why is heme present? Because oxygen levels are high. So oxygen levels are high, which means we don't want the fermentation genes on. Oxygen levels are high, heme is made, heme activates HAP1, HAP1 activates ROX1, ROX1 protein is made. What does the ROX1 protein then do? Well, it's a DNA binding protein. So it uses its DNA binding motif to reach into the major groove of DNA. It can bind with specificity, and it recognizes sequences that are found in front of all of the fermentation genes. And ROX1 turns those genes off. ROX1 incidentally stands for regulated by oxygen, which indeed this protein is. So let's think about that. ROX1 is a repressor. It's repressing the fermentation genes. Fermentation genes are repressed due to ROX1. ROX1 is around due to HAP1. HAP1 is on due to heme. Heme is made due to oxygen availability. When oxygen is available, we want the fermentation genes off. When oxygen levels are high, the fermentation genes are repressed. Beautiful system. Oxygen levels drop. The cell's going to die if it continues to respire. When oxygen levels fall, the cell can no longer make heme. If there's no more heme around, HAP is no longer active. If HAP is no longer active, then ROX1 is no longer expressed. And if ROX1 is no longer expressed, then the fermentation genes revert to being on. When there's no heme and low oxygen, the fermentation genes are on. Perfect. So this is coordinated regulation. There are over a dozen fermentation genes, and they're all regulated in this way. Every single fermentation gene is responding to oxygen availability in this way. Coordinated regulation. Multiple genes responding to one factor. Now, what that factor is really up to you to define. Some of you might say that the fermentation genes are regulated by the factor of oxygen availability. Sure. Some of you might say that it's regulated by the factor of heme biosynthesis. That's fine. Maybe you'll say it's regulated by HAP activity or by ROX1. All of those are perfectly reasonable answers. But what's important is that it's coordinated regulation because all of the different fermentation genes are being regulated by this single factor of oxygen availability seen through heme and HAP1. So that is the story that we were telling there for coordinated regulation. Now, combinatorial control. We're not going to talk about a specific example here because this pertains to a lot of genes. I'm blowing up a single gene here. We have the A of the ATG. We've got the promoter here with TBP. And let's use this opportunity for a little bit of review. What binds to the TATA box? TBP, or TATA binding protein, which is one subunit of the entire TF2D general transcription factor. TF2D recruits TF2A, and together TF2A and D recruit TF2B. Then F joins the party, but what makes F unique is that it escorts the RNA polymerase II enzyme, which includes its carboxy terminal tail. Next is going to be TF2E to stabilize this assembly, and finally TF2H, which usually is recruited back here so it can interact and phosphorylate with that carboxy terminal domain. This is the pre-initiation complex. This is the general transcription factor in RNA polymerase II complex that is needed for transcriptional regulation. We've talked about this before. What's new to the story, if I've got the color for it, I guess I'll go back to blue, is that draped over all of this is this giant multi-subunit complex that we affectionately refer to as mediator. And mediator mediates the activation of transcription. It is mediator that tells TF2H to go ahead and phosphorylate the carboxy terminal domain of RNA polymerase II. It is mediator that green lights transcription to occur. So then the question becomes, well, what's talking to mediator, right? What is signaling mediator to say yes to TF2H, go ahead and do that phosphorylation, release RNA polymerase II down the gene for transcription, or is telling mediator to say no, block TF2H phosphorylation, keep the pre-initiation complex stable, do not initiate transcription? What is mediator responding to? And the best kind of cheeky answer to that is, what is mediator not responding to? All of the signals and inputs that have anything to do with the regulation of this gene are feeding directly into physical interactions with mediator. Now, that can be enhancers and other transcription factors that are interacting with mediator by the looping out that we talked about earlier. This can also be environmental signals that have the ability to directly influence mediator. Anything that plays a role in regulating this gene is going to influence and impact the regulation of this gene by interacting directly and physically with mediator. Some of these signals are going to say, yes, activate. We need the expression of this gene. And other signals are going to be negative regulatory signals. And they'll be calling for the gene to be repressed. You can think of this in kind of a modern day real-world application. So if we're going to begin absorbing glucose out of the bloodstream, right, a very, very commonplace thing, you can imagine all of the different variables that are going to pertain to that. Should we absorb glucose out of the bloodstream? Well, if our stomach is full, we should. Because if our stomach is full, that means more glucose is on the way. We don't need so much in the blood because glucose from the meal we've just consumed is going to be released into the blood soon. Okay, that's a consideration. What are cellular ATP levels? If cellular ATP levels are high, we don't need to absorb glucose from the blood. If they're low, we do need to absorb them. Okay. What's the nutritional requirements of the brain right now? The only food that the brain can use is glucose, whereas all of our other body cells can mobilize energy from fat metabolism, from protein even. So is the brain starving for energy? If the answer is yes, leave the glucose in the blood. If the answer is no, then take the glucose out of the blood. What are insulin levels looking like? So you can imagine that that simple question, should we absorb glucose from the bloodstream, has inputs from the brain's metabolism, from the digestive system, from the cellular needs for ATP, etc., etc. And maybe the brain is saying, leave glucose in the blood because I need it. And the stomach is saying, absorb the glucose from the blood because I'm full. And maybe cells are saying, I'm low on ATP, leave it in the blood for me. And some other factor is saying no. And this gene is for a glucose transporter. This gene encodes a protein that will bring glucose in from the blood. So the question becomes, how does this gene know to express or not? The brain is saying yes, the stomach is saying no, the cells are saying yes, some other factors saying no. How does the cell know, how does this gene know, yes, express me, or no, don't? Someone has to be counting the votes. Someone has to be weighing these priorities. Someone has to be making the ultimate decision, yes, fire, we need this protein. No, do not fire, we don't need this protein. And that entity doing that decision-making is mediator. Mediator, just like a mediator in a courtroom, just like a mediator in a negotiation, is hearing all sides of the issue and making the final determination. If the yes votes win, mediator is going to signal TF2H to do the phosphorylation of the carboxy terminal tail of RNA polymerase II, and we will fire this round of transcription. We'll make this messenger RNA. It'll be capped, tailed, spliced, and exported into the cytoplasm. It'll be translated, and the protein will be made. the cell will respond accordingly. If mediator decides that the no's win the vote, then because the no's win the vote, we're going to have no firing of transcription. The carboxy terminal domain of RNA polymerase II will not be phosphorylated, will never make a messenger RNA, the protein will not be expressed. So it is mediator that counts the votes. Think back on what I said earlier about combinatorial control. One gene responding to multiple factors. Well, that's exactly what's happening here. This one gene is responding to the multiple factors coming into it, weighing into the decision of expression, and it's mediator that is the kind of the chairman of that decision or chairwoman of that decision and making that final determination. So that's what we mean by combinatorial control. So just to put a finer point on both of these concepts, the looping out as well as mediator, again, something better than I could ever draw. Here we see just the looping out idea. So we have the promoter is here, we see RNA polymerase II is here, we've got some of the general transcription factors, they're just not showing the entire pre-initiation complex. And then we have these transcription factors or enhancers that are binding far, far away from the promoter, but we're able to bend the DNA back on itself, right? We've got this big looping out occurring here, and based on that looping out, we can get these far-ranging transcription factors to interact directly with the pre-initiation complex. So that's the looping out. Now, when it comes to mediator, here's what we're talking about there. Same thing applies. We've got RNA polymerase. You can imagine right next to the promoter, we've got the entire pre-initiation complex. Draped over it is mediator. But now we're getting that bending out, that looping out multiple times. So here's a transcription factor that's weighing in and saying, yes, we need this gene responsive to oxygen. And then we get another segment of DNA looped out, so a second transcription factor can say, no, do not express this gene. We have no oxygen, but we've got plenty of sugar that we can ferment. And then you get more DNA looping out, so this third transcription factor can say, yes, we need more energy. We need this gene to fire because we have osmotic stress and we need ATP on hand to deal with that. And mediator's weighing all of that. Yes, no, yes. Mediator counts the votes, clears the gene for transcription, tells TF2H to phosphorylate that carboxy terminal tail, and down this gene, RNA polymerase will go to transcribe it, make a messenger RNA, and then the rest of the story of gene expression that we talked about last week. So this is a nice way to bring the looping out together with this combinatorial control. So combinatorial control, multiple factors affecting one gene, coordinated regulation, multiple genes regulated by one factor. So, all of those topics related quite well, right? We really built a nice cohesive story. We started with a very, very simple example of tripoperon regulation. And by talking about tripoperon regulation, we were able to talk a little bit more about DNA binding, major groove accessibility, and specificity of DNA binding. We even talked a little bit about repressors and this abstract idea of regulating at a distance. That set the stage and the foundation for a much deeper consideration of coordinated regulation and combinatorial control. All of these topics really go together beautifully into one big story of gene regulation. And now put that story to bed, because RNAi has nothing to do with transcription regulation at all. Everything we just talked about is regulating the initiation of transcription. RNAi is a fail-safe mechanism of how do you stop a protein from being expressed after transcription has occurred. So this is post-transcriptional control, which makes it a completely different topic entirely. So, if we have a mature messenger RNA with its poly A tail and its 5' cap with its weird guanine methyl group, etc., and its introns have been spliced out, so transcription has occurred. Pre-initiation complex assembled, mediator gave the all clear, the gene was fired, RNA polymerase did its transcription, and we processed the messenger RNA. This thing is now in the cytoplasm. But we no longer want to express this gene. The cell can rely on this relatively newly discovered obscure mechanism of post-transcriptional control that we call RNA interference. So what happens in RNA interference? In RNA interference, we are triggered by a fairly long region of double-stranded RNA. We're in the world of RNA now. So double-stranded RNA is pretty rare at the messenger RNA level. Now, yeah, we got tons of intramolecular base pairing like tRNAs and ribosomal RNAs, but these are two different strands of RNA that are double-stranded to one another. That's not usually seen. That's easily recognized by the cell. When that double-stranded region of RNA is recognized, a cleaving protein called dicer will come in and cut these regions up into small segments of double-stranded RNA. And it's called dicer because that's exactly what it does. It dices these molecules up. So now we have these short segments of double-stranded RNA that were processed by dicer. These are then going to be pulled apart into single strands of RNA. And these single strands of short RNA that are around because of dicer are called small, because they are short, interfering, because they're going to block gene expression, and RNAs, because that's what they are, or siRNAs. These small interfering RNAs, these siRNAs, are then grabbed by a large protein complex called RISC. Each RISC is going to grab one interfering RNA. And RISC stands for RNA-induced silencing complex. So RNA-induced means turned on by RNA, and silencing complex means a complex that silences things. What it's going to silence is this gene expression. So once RISC has bound to these small interfering RNAs, that RISC is now activated. And what it is going to do, once activated, is it's going to use those small interfering RNAs to find any other RNA molecule in the cell that is complementary to that sequence. Now, we already know, because I'm using the colors carefully, that every single purple small interfering RNA is going to be complementary to this red messenger RNA that I just drew here. How do we know that? Because when I started this, I showed you that the purple strand was complementary to the red sequence. So the purple strand is still complementary to the red sequence. These small purple, small interfering RNAs are going to base pair to the single-stranded messenger RNA, because messenger RNA is always single-stranded. But now, what's also associated with that small interfering RNA? RISC. And what does RISC do once it is recruited to an RNA molecule, such as we've drawn here? Once its little small interfering RNA base pairs to a target molecule, what does RISC do? Oh man, it annihilates the RNA that it was targeted to. It chops it up into small, small pieces. So RISC becomes the attack dog, drawn to new RNA molecules that match in complementary sequence the small interfering RNAs that RISC is holding on to. It doesn't matter how many messenger RNAs are present in the cell, we've got so many active RISCs that those RISCs will be drawn to all of those messenger RNAs and will chop them up into pieces. By chopping up all of these messenger RNAs, translation never has a chance to occur, and the protein will not be made. So again, RNAi is a post-transcriptional regulation mechanism. It's a way to stop a protein from being made, even though transcription has already occurred. There's one more part of RNAi that is present in some cell types, and it's an amplification of this system. So it might serve us well to kind of recap. We'll go back to the beginning. We have our processed messenger RNA, and it's triggering RNAi because we have this double-stranded region. So that's going to recruit what, if you remember? I think I used blue for this one. That's going to recruit dicer, and dicer is going to chop this up into small interfering RNAs. And it was at this point that I said these RNAs are pulled apart and given to RISC as small interfering RNAs. But we can also have a side reaction here. Some of these can be grabbed by a protein that's called RNA-dependent RNA polymerase. RNA-dependent RNA polymerase. This is a protein that makes RNA. It polymerizes RNA. It's an RNA polymerase, but what does it use to make RNA? It uses RNA to make RNA. It's dependent on RNA to polymerize RNA. So RDRP is going to take some of these double-stranded, soon-to-be potentially small interfering RNAs, and it's going to use that as template to polymerize new RNA in an RNA-dependent way. It's going to use these double-stranded RNA molecules to make new copies, single-stranded, of that sequence. What is each one of these? Each one of these is its own small interfering RNA, which means each one of these can be grabbed by a new RISC. It can activate that RISC. And so I forget what I drew before. I think I had 6 RISC bloodhounds on the prowl. Now we could have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 active RISCs. I'm not even counting because I can't talk and count at the same time. 15 active RISCs. All activated by these small interfering RNAs that were made by RNA-dependent polymerase. This is an amplification of the RNAi response, and it results in a much faster, more robust RNAi reaction than we could have had without RDRP. So those are the main players of RNA interference. Dicer to kickstart the process when it recognizes double-stranded RNA. RISC to implement the process. It uses a small interfering RNA to target itself through complementary base pairing to a target RNA. And then in some cell types, we have RNA-dependent RNA polymerase in the middle, taking the dicered, if that's a word, the dicered double-stranded RNA and using it as template to make many, many identical copies of new siRNAs that can then go and activate many, many more copies or more complexes of RISC. Post-Transcriptional Control through RNA Interference. This process was discovered and characterized by a wonderful researcher about 15 years ago now, and he won the Nobel Prize for it. The reason why I bring it up is because he worked at Worcester Medical School, at UMass Medical School in Worcester, Mass. So a nice locally grown hero there who deciphered this story in C. elegans, in the microscopic worm. So that's RNAi. Hopefully, these topics make more sense and are more cohesive for you. They make a more coherent story, I hope, where they're not just individual kind of stories and facts. We don't ever want that to be the case, but more a large story of how genes are regulated, most notably in eukaryotic cells, and how we keep proteins from being made when we don't need them. Awesome, awesome stuff. So we'll be implementing this information as we move on to the challenge questions later on this week, but you know what to do. Until then, let me know if this remains unclear in the dialog box that's about to pop up, or shoot me an email and I'll talk to you one-on-one by email. We'll even set up an office hours appointment or an appointment at a time that's mutually convenient for both of us. Let's make sure that you're as comfortable with this material as possible. 2 weeks from right around now, we will be in our first assessment week, so we want to just make sure that we are fully comprehending the material of this unit. Thanks so much for watching.